Well, thank you all for coming. This is a very special session, and I'm very excited about what we're going to be talking about. These people on my left are all my friends and colleagues, and you'd think that I should be able to just introduce them without this piece of paper in front of me. But since they have so many titles, I just have to get it right, and I'd never get it right without this. So, on the far left is Brian Brown. He's a director of the Icahn Genomics Institute, Associate Director of the Precision Immunology Institute, Professor, Genetics and Genomic Sciences of the Icahn School of Medicine. Next to him is Peter Palese. Peter is the Horace W. Goldsmith Professor and Chair of the Department of Microbiology at our Icahn School of Medicine. And to my left is Helen Mayberg, Founding Director, the Nash Family Center for Advanced Circuit Therapeutics, Professor of Psychiatry, and I have enough Icahn here, Icahn School of Medicine. But if I didn't say Icahn all the time, Carl would get very upset. So let me begin with getting you to know these people. I'm going to ask them, why did they get into this field? How did it all happen? Give us some background. And let's start at the end with Brian. Go ahead.
Hi. So I started I finished high school early, and I worked as an ambulance attendant in between high school and university and loved it. It was incredibly rewarding, and so I entered pre-med. And then it was very interesting, and I spent a summer also in Israel and working at Magen David Adom on the ambulance there. Again, loved it, but I was very frustrated by the fact that we were always treating patients, and when you work in the ambulance, you're seeing people really at their sickest and towards the end of life. And I went back to medical school, and I said, I don't want to be treating patients, I want to be curing them. And the way I'm going to cure them is by, how am I going to do that? And I happen to love science fiction, and I thought the way to cure them is to treat them at the very cause of the disease, which is in many cases genes. So I decided I was going to do research on gene therapy, and I switched tracks, and I decided to do a PhD to study gene therapy, and I've been doing it now for 24 years. I've been working in the field, and that's how I got started.
Peter?
So the question is, how did I get into what I'm doing? Ask me something easier, Dr. I'm a damned foreigner, okay? I stepped off the boat some time ago, not knowing anything, and I'm very thankful. This was my first real job, Mount Sinai, and so I think I almost beat you, David. Dr. Davis said I'm a long timer at Mount Sinai, and I basically slipped into what I'm doing. I love what I do, and I think I hope we are successful, and I love what I do, and I just want to keep going.
Okay, Helen? Tell us about how you got into this.
So I got here by accident too, and I think that's what all scientists doing innovation do. You don't have a plan and you end up in it. So I grew up in a household with a general practitioner father who also went on to actually build some of the early physician-run hospitals in Southern California, and had an uncle who was an amazing physician scientist. And I really didn't want anything to do with it. I wanted to be an artist or a writer, but I was really good at science and math. And as I went through and realized to be a real creative in art is a real arbitrary. And there were issues in college. I fell into psychobiology as the early days of neuroscience. And as I learned about abnormal behavior, it was pretty clear that we didn't know anything about the brain. We made up and described it and thought we knew what we were talking about, and tried to avoid medical school, but decided that was the only way I could do it. And when I got to medical school, I was a troublemaker because I could not stand how psychiatry was taught or thought about, because it didn't consider the brain. And at that time, we actually started to have tools where you could map the brain. And I was hooked, but I decided so I'm not a psychiatrist, sorry. I'm a neurologist. And so the issue of psychiatry was horrible because I hated the language. Sorry again. But that neurology didn't care about behavior, and I cared about the intermixture of behavior in the brain. So how do you understand something like depression from that point of view? So I wandered around a long time, trained in neurology, trained in brain imaging, how to map brain receptors, brain circuits, structure, function, and then actually fell into depression and have been there ever since.
Great. All right, so now let's get down to some real stuff. Tell us what your lab is doing. Brian?
So I work on, I'm an immunologist, and I'm also work on gene therapy and genetic engineering. And my lab, since I started research, has been trying to fuse the two, which is actually similar to what the COVID vaccine, the mRNA COVID vaccines are. And so what we are trying to do, our major focus is trying to target these therapies and develop ways to, a lot of what we work in is trying to use gene therapy, DNA and RNA therapies to teach the immune system how to target cancer cells and kill them and eradicate tumors. So that's the basis of my lab in a nutshell.
Peter?
Yes, I started really working on influenza viruses when I came to Mount Sinai, and have really stuck to influenza viruses in all its different manifestations. I was working on vaccines, on antivirals, on genetics, on surveillance. So I have a real very broad interest in all things influenza. And it's still very, very interesting after many, many years working on it. And when we passed the 2019 to 2020, when COVID-19 became a real issue, I thought, oh, I know everything about influenza, this is easy. It is not. It is very, very different. Yes, it is an RNA virus like influenza, it has RNA inside. It is a respiratory virus, it infects respiratory cells. It is a virus which has a surface of spikes on the outside. I thought I knew everything. It is really very, very different. And those of us who have heard the talk by Dr. Putrina in the morning about long COVID, this is unbelievable. This is really something we should be, I think, afraid of, and I hope we will be able. Right now it is called a Center of Post-COVID Care. I hope it will be called a Center of Post-COVID Cure.
Go ahead.
So the lab is actually quite broad, the center, because I came to Sinai about 4 years ago to take what we'd been doing in depression and ramp it up. But what I really do is we map out where in the brain depression lives. So to think about it, your brain not as a bowl of soup, add some serotonin, stir, but that actually you have areas of the brain talk to each other. They're wired together. And as was been described, 100 years ago by Hebb, what's wired together fires together, which means like any kind of orchestra, all of the instruments in one section have to play together, and they have to communicate and listen to instruments in another part of the orchestra, and somebody has to conduct. There is no conductor in the brain, but everybody has to play together. And what goes wrong in depression is what we study. And it turns out that not only does depression localize to a distributed set of brain regions that don't talk to each other correctly, that how medication works and how therapy works is not the same, and that we've mapped to know that people who need medicine, they don't get better on therapy, and people who need therapy get therapy and don't get better on medicine. And there are people who, despite our best efforts and our best evidence, don't get better on anything. And that's actually what my lab focuses on. All of the work to map out, not that one size fits all, but there's not an infinite number of ways to treat people any more than what Brian and Peter talk about. There's precision. And what we do is we actually have leveraged technology where we implant in the brain a pacemaker. And by knowing the wiring diagram, we can drop an electrode with millimeter precision into the brain, attach it to a pacemaker, and tune a depression circuit and switch people out of a depressed state, and then they stay well. And that's what we work on. So A, it works. We now have to figure out who are the people that are best for that. We spend our time trying to understand how it works, how to actually integrate once your brain is working, how you actually use therapy, and how it doesn't treat all components of depression. So, the big picture is how to take technology, not be afraid of it, but actually with evidence, implant with precision that doesn't have the kind of precision of a gene or a particular virus or a particular molecule, but to take what we know and to help people. So all of the work is directly impacted on patients are our collaborators, and how do we move forward from there?
So let's talk about breakthroughs. Let's talk about what's happening in your lab that you think have breakthroughs that are really going to change medicine over the next decade or so. Brian?
Yeah, so I mentioned before, the problem I work on is how to cure cancer, right? So the thing that we work on is how to target these RNA and DNA therapeutics. And I like to say, if you solve targeting, you solve cancer. And what I mean by that is, we can take cancer cells in a petri dish, I can put chemotherapy on them, and I will kill every single cancer cell. And I would do that, I could wipe out someone's tumor, no problem, with enough chemotherapy. The problem is they'd be dead before their tumor would be dead, right? So targeting is absolutely essential to making these therapies work. If you think about, if you have a pain in your elbow and you take a Tylenol, that medicine goes everywhere. It's not just treating the pain in your elbow, but if you could get it there and only work on the cells that are causing the pain, or those chemotherapies only killing the cancer cells, well, you could wipe them out. So what we can do, that's very hard with traditional medicines, but with these new wave of RNA and DNA therapeutics, we can put sophisticated controls in them. And so one of the things that I invented is a way that we can target RNA and DNA therapeutics to only activate in certain cell types that you want them. So we are working on making those RNA and DNA therapeutics only kill cancer cells, but when they get inside your healthy cells, your cardiac cell, your heart cells, your lung cells, they're not going to kill them. And that will allow us to eradicate the tumor without causing any sort of toxicity to the patient. And so that's what we're working on, and we have invented something to do that, and it's already in clinical trials, and there's several different waves of therapeutics that are being developed around this principle and this targeting concept.
So give us a little more. Tell us how long you think it could be before this is really going to change the lives of people. How optimistic or pessimistic can we be? Because all of us who've done clinical trials know the vast majority of them fail.
Yeah.
So should we be optimistic? When do you think we'll have something to count on?
So, I'm always optimistic. I think no matter what, it's rare that they fail. So they may not work, but then we learn something, we bring it back to our labs, and then that allows us, I think of myself often as a bioengineer. So what I do is when the clinical trial doesn't work out the way we think, we then, we have data from that trial that tells us, oh, you know what we needed to do? It turns out that we needed to add this switch to the drug, or we needed to add this. And so I think, it can be slow, but then once it works, it's like a big bang. All of a sudden, you have many, many different drugs that can be created around that principle. And that's what we're seeing with these mRNA therapeutics. It's not just going to be used to treat COVID. It's going to be used to, create potentially flu vaccines, cancer vaccines, all types of things. So I think it's hard to give it a timeline. I think the other thing that kind of slows these timelines down, especially in cancer, is that we usually when we start these therapies, we treat the most sick patients, the people who have the most aggressive disease. And this makes it much, much tougher to have success. But then once you have success there, we start to give it earlier and earlier, and then it becomes even more successful and more effective, the therapy. So I'm optimistic.
So Peter, two related questions. You've been working on a universal flu vaccine for a long time. We all know that every year we need a different flu vaccine. What you want to do is give us something that we can take once and it's going to work. Tell us how hard that is, how far along you are, and then transition into, is it even tougher for COVID? And when do you think we're going to, with all these mutations that happen, will we always be behind on COVID?
Dr. Davis put down the problem with influenza. It is changing every year, and there are actually 4 components. It's not only one influenza virus, there are actually 4. So making vaccines requires making a new formulation every year, and that is not easy because we don't know which direction the virus goes. And we have, I think, good vaccines. And everyone sitting here, please take the influenza virus vaccine. The vaccines are better than their reputation. However, no, they are really much better. And I will explain why that is. However, it would be nice to have something which you don't have to take every year, but rather once every 20 years, or even for a whole lifetime, like measles, mumps, rubella. So that's what we are working on. And we are trying to make a vaccine which induces antibodies, when given to us, against the conserved portion of the virus. So we are trying not to make a vaccine against the specific 2023 virus, but rather a conserved one which is shown by all influenza viruses. So that's what we are trying to do. We are in human trials, and I think, to be quite honest, we were sort of interrupted when COVID-19 came, because the attention, and rightfully so, went towards COVID-19. So we hope that if enough interest and enough resources are being spent for influenza virus vaccines in the future, I think we can be faster than we have been in the past. Now, the situation with COVID-19 is different. This is a wonderful vaccine we have. It's a major breakthrough, the Moderna and Pfizer-BioNTech. I don't think we have ever seen anything in the last decades, I can say. It is something which is fantastic. Went from 10 years, usually the development time, to 1 year. It is a very, very effective vaccine. I think come next October, first week of October, I think people in Stockholm will show their also admiration for this fantastic medical breakthrough. I think this is a really fantastic breakthrough in terms of having saved many, many millions of lives all over the globe. Nevertheless, there is some niche for improvement. One, there is something of what is called cold chain. That is, from the place where the vaccine is made to when it is being given, the vaccine, as we know it, the mRNA vaccines, have to be kept at -80 centigrade. So this is what is called the cold chain. Very expensive company. Every minute it has to be at -80. So it would be better to have a vaccine which is more stable at room temperature. And the other one is that we all know there is a breakthrough happening right now. People who have been vaccinated 3-4 times still come down. They come down with a more mild disease, I believe, but still, I think there is some better mousetrap to make a vaccine which prevents really infection and thereby transmission. And that's what we are trying to do at Mount Sinai. We have 3 different laboratories. It's Adolfo Garcia Sostre, Florian Kramer, and myself. We're all at in the microbiology department, and we have joined forces to make a vaccine which can be manufactured very easily, one which has a very easy cold chain. We don't need -80, we need 4 degrees centigrade. Every kitchen refrigerator has that. And we have a vaccine which is, can be given through the nose. So that means the virus comes through the respiratory tract, and if we vaccinate through the respiratory tract, we get a better mucosal immunity so that the virus cannot really take very easily. So an intranasal vaccine should have an advantage in terms of preventing infection and also preventing transmission from one person to the other. And so we are working on this, and we hope that we will find that there is a niche for such a vaccine in terms of having a much easier cold chain, having the ability to give it through the nose and induce what we call mucosal immunity, preventing infection and also preventing transmission from one patient to the other. And then there is a third issue that we can make very easily new vaccines with these variants of concern, VOC. There is maybe not another Omicron, but there may be something, give me another name. We don't know whether this will be continuously changing, and there our vaccine approach is also a very easy one. We can easily flip different variants of concern into this. So I think you asked the question, how long will it take? I think you have to ask the virus. Yeah? The virus is going zigzag constantly, and I think it is very difficult to really predict it.
But can I add one thing?
Go ahead.
I just want to just say, one of the things that's interesting, because Peter is the chair of microbiology. I'm the associate director of immunology.
Icahn School of Medicine.
Icahn School of Medicine. But we actually, Mount Sinai is a very unsiloed place as opposed to many places. So Peter and I work together actually a lot. And, I'm talking about developing targeted therapies for cancer. We actually, we use each other's things all the time. And this is, I think, where the big bang breakthroughs can come through, where we cross-pollinate each other. He's working on something for, virology. I'm working on something for cancer. And then we say, well, maybe that would actually work better for cancer, and maybe what I'm working on will work better for flu. And this is, I think, the type of interactions we have all the time. Actually, Peter and I have published together, and I think this happens all the time at Mount Sinai. And that's where I think the next breakthrough.
Science paper.
Yes, science paper.
Right. So let me ask Helen to get very granular. She told us lots of people get depressed, and there are lots of different kinds of depression. How many people are actually going to want to have a deep brain electrode put into their head to cure their depression? How do you figure out who should get it? And have you done it? Tell us your experience, and keep it as granular, as patient-centric as you can.
Okay. So I do want to get back to something, because
Sure.
one of the amazing things about synergy and big science and innovation is, as I'm listening to two colleagues that I actually met for this meeting. They weren't colleagues, I only knew about them. As I listen to Brian, I'm already getting an idea about the same kind of precision that goes into its location, location, location in the brain. He's got it at the gene level, at the protein level, but all of medicine is about precision. And it just kind of depends on how much we know. We just don't know as much about the brain, and everyone hears that and is impatient. But we know actually lots and lots about the brain at the molecular level. We know lots and lots at the clinical level, and how we bring them together is the issue. So that gets to your question. So shockingly, I think more people have true treatment-resistant depression than we know. Everyone's aware that the numbers now, particularly in the lockdown with COVID, hasn't caused depression, but it's actually unmasked depression in many, many people that were vulnerable. And the numbers are astronomical, and it's frightening. And all of those people, regardless, you don't need an electrode, need to go for help, treatments work, but having the brain under stress is damaging these circuits. And what we're trying to learn is people's brain, when they can respond to therapy or a drug with a first episode, is in a different state than people who stop responding. And there is almost a malignant degeneration that happens to some people, about 10%. So if 10% of the country, of the world, has diagnosed with a major depressive episode, and 10% of those at some point go on to have treatment-resistant depression, everyone in this room, if you haven't had a depression, someone close to you has. And I'm not asking for raising of hand. It's just incredibly common. And the issue is, everyone also knows people that stop responding. That doesn't mean the treatments don't work. It means, like in cancer or in something, we're temporizing, we're not curing. So it's a big problem. And you're right, who in their right mind would want a neurosurgeon to drill a hole, even a small one? It's brain surgery, it's not getting your nails done. You have to drill a hole with precision, millimeter precision, but there are neurosurgeons who do this every day for Parkinson's. So think about this, Parkinson's disease, you're losing your dopamine cells over time, You take a medicine that replaces it. Soon the medication itself gives you problems, and you have very little time during the day when you feel good. And you can drop in an electrode because scientists figured out the motor circuit that responds to the dopamine, and you can implant, and people can be well even though they still die of Parkinson's disease, and they have a better life. All we did was leverage that technology, but you can't stick the electrode willy-nilly. You have to have precision again, and we have precision. So again, we do small studies. It's research generated. How do we get it? I can tell you right now, in our lab, we have NIH grants. We now have prototype devices. I can measure where we're stimulating off the brain, and I can get a brain readout that's telling me if it's working or not. And my team has figured out the signal to actually know that you're at the finish line or not, because life happens on top of depression. And how do you discriminate having a bad day from actually having relapse? So we've got it implanted. I've got people, we started in Toronto, 2003, the first case. I've got people that write me every year with a Christmas card of how they're doing, so that this isn't like other treatments. We can get through a clinical trial, you're well for 6 months, everybody's had that experience, and you relapse. This is like you have a heart arrhythmia. You correct the arrhythmia, and you then live your life. You have ups and downs, and we have people chronically implanted now 15 years. So it's there, but not scaled. And so the issue is, look, you can come to me, I can give you the number, but in fact, we can't take care of the community that needs that. We can do small experiments. We have colleagues that are taking different strategies. Industry has wanted to do this and has tried, but the details need to be worked out, and it looks like they're going to try again soon. But in order to do it well, there's the precision of the location, and then there's the million-dollar question: how do we match patients to the treatment that's best for them? And that's true whether you're deciding on a drug or ketamine or psilocybin. One treatment does not treat anybody, and don't let anybody tell you that it does, if you take nothing away from what I'm saying. But that trial and error is how we do it now. And there is this where you get to this point that this may be the future. We use this in very bad obsessive compulsive disorder, very bad Tourette's syndrome. It's being examined for Alzheimer's disease, where the circuit is a little harder. It's like that any illness where there's a wiring diagram that you can find is broken, you have potential for this technology, which means you also might be able, what's the future? Getting this scaled, but more importantly, to understand how it works. If you need to have an implant to stay well indefinitely, I'll take it. On the other hand, not everyone would still have access. How can we learn what we learn? Technology has made it to where over the last 5 years, I can measure the brain. It can record, and I can beam the information and look it out on my desk, to actually getting to where maybe you don't need to have a wire with a implanted battery pack, that you can do it externally. Or maybe I can use something that Brian's developed to have a gene that turns on receptivity, and you can just actually get precision outside the brain. I just made that up, but that everyone knows you can do magnetic stimulation outside the brain. What we do is for people that fail even transcranial magnetic stimulation, because if the connection is broken, you can scream however long you want and loud you want in your own house at your own phone, but if it's lost the connection to your neighbor, nobody's going to hear you. So that's kind of where I see this, and that's what I don't know. But the scans are, kind of to say one more thing on that, because it gets into digital health. The scans are helpful, but again, getting a scan to decide how to treat, not quite ready for prime time, or to scale it. On the other hand, we're starting to look at your face is driven by brain circuits, and the same brain circuits that break down in depression are involved in emotional expression on your face. So we're starting to look at, when you talk to someone who's depressed and they don't quite look right, it's different from somebody sad. that actually that's a reflection of a broken brain circuit, and maybe we can use the scan to help us to just read the face with AI and make the diagnosis with AI without the scan, but all starting with the brain. Look, Einstein said it should be as simple as possible, but not simpler. And the same is true here. We do the science with big teams, and then we look to simplify and scale. And that's what we need your help for. So
I'm going to ask each of you to just give me a very succinct 1-minute answer to this question. Fast forward 10 years. What's it going to look like in each of your fields, and how will it be affecting real people every day? Brian?
I'm coming at this from an immunologist point of view, but I think in the past 10 years, our ability to drug the immune system has been, monumental. It's changed the course of rheumatoid arthritis. It's changed how we treat cancer, and I think we're only getting better at doing that. And I think we're finding that almost every disease has an immune component, and we have, for the first time in medical history, the ability to drug every gene. Okay, and I say that because RNA and DNA therapeutics allow us to target every single gene in the genome. We couldn't do that with the type of things, the type of drugs we've been taking. Even antibodies can't drug every gene. So what do I see in the next 10 years? Well, I see that as we're learning the causes of each of these diseases. Not necessarily the cause, but also what's driving those symptoms. And we are just going to continue to build therapeutics that target these pathways and that can either dampen them in the case of too much immune response, or enhance them in the case where you want to boost an immune response, like against cancer or against a virus. So in 10 years, we will have much better control of many different diseases because we will be drugging the immune system in a much more precise way.
So to make it really simple,
yeah,
we take antibiotics for infection. They generally work. It's been a revolution in medicine. 10 years from now, because of the work that you and others are doing in genomics and immunology, will we be able to say the same for cancer?
Absolutely, yes, no doubt. I mean, what happens is we transform a disease, and then overnight you sort of forget that the disease is, that 5 years ago, we were in a different place at how bad it was. I mean, if you look at, even something like melanoma or many different, rheumatoid arthritis, people used to be in a wheelchair. We now give Remicade or Humira. Those people can get up and walk days later. And so there will be, for each of these diseases, breakthroughs that come, and we will transform them through these new medicines. I mean, really, for me, one of the most exciting things about the COVID vaccines, besides the fact that it helped to end the pandemic, is that it gave us an entirely new form, a new platform of medicine, and we can just build on that for treating many, many different diseases. And it may be me, but it may be someone in the lab beside me who says, hey, maybe I have discovered this gene which is causing disease X, and I never had a way to treat that gene. Wait a minute, now all of a sudden I can do it because there's mRNA therapeutics out there. And that's going to be transformative. It will, it always happens like that. We'll see an exponential growth of how effective, of how many different diseases these treatments can treat.
Peter?
Yeah, I echo what Brian just said. I want to quote Joshua Lederberg. He was one of the youngest, or the youngest Nobel Prize winner, and he said, microbes outnumber us. We have to outsmart them.
And we will.
Exactly.
Succinctly.
So I'm an optimist, but a pragmatist. And I think that in 10 years in my field, if we can get this to where it's generally available, that's a win. These trials take a lot of time. We can't get impatient. But in fact, like in every other field, when I started, the idea of saying depression was a circuit disorder, people laughed. And because I was a neurologist, they ignored me. And now that's an organizing principle of all psychiatric disorder. And so that you can't, foresee the future, but you actually can watch a trajectory. And I think the fact that people are coming back around and seeing that if people stay well, that maybe an implant isn't actually as distasteful or inappropriate as we might have seen it. Because if you can do something that can get people well and keep them well in the prime of their life, it's like Parkinson's. If you wait until, we took the people that are at the end of the line, generally the time that it works for them, what would have happened if you had started not when they were 50, but when they were 35 in the prime of their life? And I think we have to kind of address the brain, address evidence, and not be afraid by the past of a belief system that wasn't supported by data. And I think that's where we're going to be. We're going to have less stigma, and we're going to approach these with treatments that are evidence-based.
Okay, so we have time for questions now. We have a mic. It's mobile. So put your hand up high so we can see it. Go ahead.
Hi, I'm Jakub Hlavka from USC Los Angeles. I'm curious about what you just said about kind of catching the disease early. I work in Alzheimer's disease, and I'm curious if there's potential in cancer or other fields to really do screening much sooner and then potentially achieve greater efficacy than catching it too late. And also potentially using these technologies, which today might be high cost, high risk, in patients who don't have significant clinical symptoms, but might actually benefit the most from these early interventions.
Is that a question? I mean, I think these things are coming together. For sure, if we can cure it early, if we can treat it early, I mean, this is, first surgery if you can cut it out, but then, if you can catch a disease quicker, then you can give less drug, and you have a much better chance of success. And the screening platforms are getting much, much better in terms of our sensitivity to pick things up earlier. It can feel like we're not there yet when, right now we may not be in the moment, but it could be 1 year, 2 years away. And I think, if there's another lesson to take away from COVID is, put enough infrastructure into something like we did for the vaccines, and a seemingly intractable problem becomes tractable. And I think that's the case here as well.
Over here. Go ahead.
Brian, this question's for you. We've long known that cancer is pretty clever and finds a way of mutating, so what is effective for a short time becomes ineffective. So when you talk about your 10-year horizon and the paradigm changes through these new technologies, how do you bake in the fact that cancer cells can morph?
Yeah. So I would say it's an interesting thing, dichotomy between immunologists and, well, not really, but the immune system, and this is an amazing thing, evolves as fast and can evolve as fast or faster than viruses. So one of the things that we're excited about is, yes, the cancer can evolve, but your immune system, your T cells and your B cells, your antibodies and your, the cells that can kill other cells in your immune system can evolve just as quick. That's why we can, even though we've never seen COVID before, we can still make antibodies against it. And we can do the same for cancer. There is a finite limit as to how much a cancer can mutate, because at some point, you mutate away and then the cancer can't grow anymore. There's like it loses its ability to be fit in the same way that the virus, can't evolve infinitely. Maybe Peter will disagree. Can't evolve infinitely. So we're at a point where a lot of the mutations come down to these 4 letters, the letter of the DNA molecular code, and we can sequence every one, find every mutation, and then figure out how can we turn the immune system against those mutations. So I'm still confident about the 10-year horizon. Okay. Don't invite me back, though, in 10 years.
There's a question. Kurt has a question. Over here in the aisle, in the center.
This was fascinating. Thank you. I'm a PhD clinical psychologist. So I send you a depressed patient, Helen, and you do functional MRI or whatever you do. How do you ascertain, do you do transcranial stimulation? Do you do ECT? Do you do implant? What's the decision tree process?
So there isn't yet one, but I can tell you what we have done, because we derived, so first of all, you have to set up a clinical trial of one treatment versus another. So in our example, cognitive behavioral therapy versus 2 different antidepressant drugs, randomize people, treat them. And our goal is actually like Ryan and Peter, you're in remission, you're well, or you had so little response that you shouldn't have bothered. Then take that data, having done a scan, and look to see whether or not the brain, any brain pattern, can discriminate wins and losses to each treatment. We identified that with both PET metabolism scans and with resting state fMRI scans. And then you have to do the next step, which is to say, I have my biomarker, and we did this with a grant for PET scanning. Are you a high insula person or a low insula person, a CBT person or a drug person? I'm going to treat you by brain type. And I'm going to see now for the next 3 months, did you respond or not? So actually we were able to show that we could make a prediction that was better than flipping a coin, which is what we do now. We've published on an fMRI pattern as well. We haven't yet done the prospective testing because the response is, well, who in their right mind would do a scan to treat someone? Just imagine that if you said that to an immunologist, I'm not going to get your chemical profile to define what kind of cancer type you have. So again, the stigma of mental illness is very different. So at this point, you actually have to do the experiments that compare, because someone will do a study on ECT and say, I know a good or a bad for ECT. There's still the element of someone making the decision. But what we have done, we have data from therapy responders, drug responders, ECT responders, ketamine responders, brain stimulation responders. And we've looked at the same patterns of connectivity, and we've now derived what's the one where the system totally breaks down.
So do you see in the next 10 years, Ken's question, that process being compressed where it can have real functionality?
I think so. So here's my 10-year horizon on that, given that I could get the work done and I had access, is that you're ill. I determine right now, I'm going to see you twice, and I'm going to look over time to actually see if you're progressing. Because, depression is about something goes wrong, we don't know why. Of course, I'd like to cure it and predict it. Good luck. That's going to take too long. But I want to know when you actually can no longer adapt. And when you lose adaptation, that's when we make our decision. And I think that what I'm seeing now is you shouldn't have to wait to fail 4 drugs, evidence-based psychotherapy, ECT, lose your memory, to say, okay, now we'll put an implant in you. I'm operating on people now that are 30, 28, who actually have lost their 20s. We can't afford to have people lose the best times of their life because we're afraid of a treatment when we operate on every other part of the body. So I think we have to do with colleagues like you, the heavy lifting of how to get the evidence to support the notion and not just kind of dream about it. But that's kind of, I think, the process.
Yeah. Elliot.
Thank you. I was impressed with your statement, Peter, at the beginning about chemotherapy works. Has anyone working on isolating cancer cells, finding specific markers, develop antibodies against those markers, and then tag the antibody with something like radioiodine or specific chemotherapy? And if so, sounds like it should work.
Yeah, the answer is a great question. Yes, absolutely. We call them antibody drug conjugates. You take an antibody, you put like a poison, but actually you take, radioisotope and you deliver it. As was mentioned before, the cancers do evolve. So that can be a challenge to find the right molecule, the right receptor on the surface of the cancer cells to target it there. So that still is a challenge of how you target it to every last cancer cell. And I think the success will be achieved in not just trying to use one antibody, but use many to deliver it there so the cancer can't evolve around it. I'll also mention that some excellent people in Peter's department are trying to exploit viruses, their ability to evolve as fast as cancer to create what we call oncolytics. So these are viruses that will only get into cancer cells and then basically just kill those cancer cells. And we hope that they can evolve at the rate of these cancer cells and beat them.
Over here. Yes.
It's probably for Dr. Mayberg. Tinnitus, depression, circuitry. What work has been done on that? The connection of tinnitus,
Oh,
sound in the ear?
sure.
Yeah.
So that people have tried to look at what brain connections give that reverberating sound. And because a lot of people with tinnitus also get depression, there was even some work in Buffalo thinking about could Area 25 deep brain stimulation, which we do to depression, actually impact tinnitus itself. So, again, that's a different circuit, and people have tried to look at how might you actually tune and don't really know what is the source in the brain of that abnormal signal, because a lot of it is also generated by the inner ear itself and amplified in the brain. So again, unfortunately, the circuit for it is less well understood in the brain, ergo then it becomes a fishing expedition to try to figure out where would you implant and is that worth the risk? But it's a really important and hard question.
Yes, other questions? This lady over here.
Hi, thank you for all your work. This question goes to Dr. Mayberg. So I know, new to me is like the new research on gut health and how it impacts the brain. Do you do any kind of integration of that research into your, or in your lab?
So again, as a neurologist, I can claim, look, I know the wiring diagram and then I hand off everybody. But what we've learned is, as you fix the brain, if you will, patients have an expectation that you fix them. And as we all know, life is about ups and downs and learning, and your brain has to be in a position to be able to learn. And that's what really treatment-resistant depression is. All we do is correct the balance of these circuits, and now actually you have to retrain to use where you are. We've actually even found that, like any intervention in Parkinson's, you can treat tremor and rigidity with a classic deep brain stimulation target, but people still freeze because that circuit is complemented by another circuit that isn't targeted. And what we've learned is that when we can get rid of that painful, agonizing immobility of depression, like I want to do something, but I got a force field around me, I can't move, and when I get there, I don't enjoy it anyway, that goes away. But if people still ruminate about the past, we don't touch that, and that's why they go to therapists. So our teams have to incorporate therapy, a good psychopharmacologist, because you have to be able to integrate the balance of all of this. You just change the state, and now you're in a new place. And part of that also becomes, what is the internal milieu? How do you reduce stressors? How do you deal with inflammation? I mean, inflammation is a growing area of concern in stress biology, as well as, the microbiome, the stress biology, and how that impacts the brain. And it turns out the areas of the brain that actually mediate stress response in the body, the adrenal gland, are the very same area that are involved in your intentionality to move. They're very close, and they're right in the circuit that we target. So we're starting to make measures of how do we impact inflammation or not. And in people who have inflammation as well as depression, it can look like the same thing. You get rid of part of it, and now you've got to treat the rest. So you must deal with the whole person. You can't just send them to a surgeon and bring them back. What we've built at Sinai, which quite frankly is the only place like this in the country, I'm not just saying this because I'm next to my big boss, okay? I never see him. But that we were allowed to bring into our neurosurgical milieu psychologists, psychiatrists, neuroscientists, engineers, with the neurosurgeon. Everybody lives next to each other, so we can riff off each other and then realize, oh my God, we really need to bring in this person because, gee, we all did our job, and instead of saying, I've handed you off, actually everybody works together to realize there is no quick fix when it comes to the brain. Just like cancer, you make a change, the brain adapts. It's really quite remarkable that with this target, you can hold it in a position and it still adapts, but it adapts in a more kind of adaptive way, not maladaptive way. So I'm totally with you, and if we didn't do that, we wouldn't be doing our job.
Last question. Go ahead. Yes, in the blue shirt.
Are you worried about the anti-science sentiment in society, even at the congressional level, and how it'll impact the breakthroughs you want to do? It could impact funding or other things, but that strong sentiment might harm your work?
Well, you can't not be concerned. The politicalization of science is frightening. When you think that something as straightforward as everyone getting vaccinated for COVID, which is, the pandemic of our lifetime, should become politicized, it's frightening. So yes, I worry a great deal about that. Brian, you are nodding.
Yeah. It keeps me up at night, but I mean, I remember going, I don't know if I don't want to go into it, but during the height of the pandemic when Mount Sinai was full, I mean, the ICUs were full, every doctor was being, no matter what you were doing, you were being repurposed to help the patients. Going down to Union Square in Manhattan, and there was like guys with bullhorns telling you COVID is fake and all this type of stuff. And I'm like, this is crazy. And going up to them and saying, go up to Mount Sinai, you go to the ER, you go to the ICU and tell me that it's fake. Didn't care. They didn't care at all. They doesn't matter. And it's poisonous, this type of attitude. And I don't understand, facts don't lie. The drugs work, vaccines work, and yet the people don't care. They'll tell you, they politicize it. And the worry is that it's not just, the COVID vaccine is one thing, we're all taking it. We're going to be protected. But what happens when these people keep people from getting polio vaccines or getting, other vaccines? What if smallpox comes back and they tell us not to take smallpox? It's a huge danger. I mean, I think it's a big worry, and we have to fight it tooth and nail, and I don't know, with everything we can do.
Peter has the last word.
Yeah, sorry.
And I just want to say something positive about the difficulties we all face. The advances in the last 10 years were tremendous. And again, if I just talk about microbes, I think we are closer to a vaccine against HIV, against malaria, against all kinds of diseases because of these major breakthroughs. So I think there is some other side of the coin that I think the future does look better than the past.
Thank you.
I like that.
Thank you all for coming.